Next speaker, hard act to follow, is Marc Pellegrini. Professor Pellegrini is an infectious disease physician. His interest is host pathogen molecular biology, and he's currently the executive director of the Centenary Institute. He's an internationally renowned scientist. His research is focused on the development of novel therapeutics and interventions for the management of a range of infectious diseases. And these are diseases that really contribute to global morbidity and mortality, including diseases such as HIV, Hep B and tuberculosis. So similar interest to what Anushka used to have. Marc, welcome, and we look forward to hearing your talk.
So a caveat to begin my talk is that I'd very much like to provide you with profound wisdom, but unfortunately we're all very different, so what's relevant to me might not be relevant to you, but what I will be able to do is give you some anecdotes along my journey as a clinician-scientist. I work at Centenary, which I think is a phenomenal institute. I've only been here for about a year or so. Before that I was at the Walter and Eliza Hall Institute in Melbourne. But at Centenary, it's an amazing place to have scientists and clinician scientists working together. So we do have very, very hardcore fundamentalist scientists at the Institute, but we also have some of our best scientists are actually clinician scientists that are still continuing to do clinical work. So we sit between the University of Sydney and RPA Hospital, so we're sandwiched between those two, and I've said many times that I think that the meat in the sandwich is better than the two pieces of bread on either side, and I maintain that. But I think the wonderful thing about Centenary is that we do our core business is discovery biology, making discoveries that are for human health. So I've called the title of my talk The Road Less Travelled, which I think is very real for a clinician scientist. There's not too many of us, and particularly when you're looking at people that want to do more fundamental science, it's not often that you'll get clinicians following that road. So I put there, try it, and I'll tell you why you should try it, is because there's no mistake. Like we said, there's never a good time or a bad time, just try it. It's only going to take, 3 years out of your life, which is pretty small. So what I'd like to do is tell you a little bit about my story and why I chose to be a clinician scientist. So I started off, did medical training, sort of was wondering what specialty to do, but decided to do infectious diseases because I spent about a 6-month stint in Uganda. So I was there at a very remote and rural hospital near the border of Rwanda, and I predominantly did paediatric infectious diseases whilst I was over there. I was actually fully trained. I was just at the end of my career, but I was as good as anyone else there at the time. And being there for that period of time really made me very, very interested in improving health and particularly treating infections a lot better. So when I started my advanced training, I decided to do infectious diseases because I had an interest in immunology, had an interest in microbiology. Actually, they were my favourite subjects at uni. And God forbid, I also absolutely loved biochemistry and molecular biology. So I thought, put them all together, surely they fit together into infectious diseases training. They don't really, so it was good, but I found that when I started to do infectious diseases and that initial training, it's very prescriptive. You follow a recipe, you see a patient, you check all the symptoms and the signs, and pinpoint what it might be, and then you treat it. And sometimes you can't treat it. So although I really enjoyed infectious diseases, I thought, well, surely there must be a little bit more that we can do. And that brings me to the next slide. This is a fellow called Jeff McCorn, who's at Centenary, and he's actually a gastrointestinal physician, a gastroenterologist, and does liver transplant physician. So that's not me. But I think Jeff probably followed a similar path that I did. But sort of I did have a desire to do things a little bit better. So particularly with infectious diseases, so many of our treatments, like you take penicillin or flucloxacillin or anything, and basically it's all based on historical data. So before this antibiotic, people died. You give this antibiotic, people don't die. But there was no double-blind randomised clinical trial to test it. But as things went on, it became more and more important to do double-blind randomised clinical trials because we were changing, swapping, combining antibiotics to understand what would be best. So is flucloxacillin best or is rifampicin best to treat osteomyelitis with a metal in the hip? So all of these things became pretty important to us. So that's clinical trials and also epi. But I was much more interested in a passion or a real fascination to make discoveries, really basic discoveries, which improve human health. It's always nice to be the first person to see or identify a biological process which might have relevance to human health. And the other reason why you might entertain going down the route of becoming a clinician scientist is because it's really shifting from following practice to actually creating practice. And I think, I was going to say, you mentioned that in your talk, and I think that's really important. And I sort of touched on it from a little bit of dissatisfaction around prescribing antibiotics to really wanting to understand, well, is there a better way to do things? How can we cure infections better than what we're doing? And ultimately, yeah, I'd love to cure a disease. Not really close to doing that as yet, but you know, that's what we all would like to do, is really to participate in curing a disease and getting rid of a scourge in our society. So most of my research is around a field in science or biology called apoptosis. And so when I started to do my PhD after I finished all my advanced training, was a consultant, and I decided, well, okay, now's the time to do my PhD. And at the time, apoptosis was taking off. It was huge. And fundamentally, it was thought to be important for human development. And fundamentally, it was thought to be important in the control of cancer. And I had a very different idea at the time, because I went into a lab that was very, very cancer focused. And I had this idea that probably the most likely scenario is that apoptosis actually developed to protect us from infections. So that was obviously a very biased view on my part, but in actual fact, it turned out to be correct. So the whole reason why we have apoptosis is not to help us get rid of the webbing between our fingers, just cut it open. You don't need a new biological process to do that. And for the most part, a biological process doesn't evolve for a disease that happens in older people after their reproductive lifespan, cancer. So the most likely scenario, and indeed now we're accumulating quite substantial evidence, that apoptosis is probably one of the most innate mechanisms our body developed to fight intracellular infections. And from the brief that you got, you'll understand why that's important to me, because HIV, hepatitis B, practically every single one, all viruses, and a lot of bacteria, intracellular organisms. So how would you get rid of those organisms? Well, you can try and find them, but ultimately this commitment to undergo suicide as a mechanism for a cell is actually a very, very good idea, because you'll kill the cell, but apoptosis makes sure that all the contents of that cell actually dies as well. So it's sort of a very altruistic way of getting rid of infections. And so that was sort of a bit of a philosophy that I had, which really, really wanted me to pursue research in apoptosis and understanding how it pertained to infectious diseases. So I went to the Walter and Eliza Hall Institute at the time and met an amazing fellow who was very, very much at the forefront of apoptosis research. And Suzanne Corey and Jerry Adams also took me under their umbrella. And so that's where I started my career. And originally, it was very much more geared towards cancer, but I said very quickly that that's not my interest. And fortunately, they were adaptable enough to sort of take me on with my ideas, and so that's where I started that pathway into understanding how apoptosis plays into infectious diseases. And from there I went to another oncology lab, this time in Toronto, and I'm not an oncologist and I wasn't that interested in cancers. But anyway, I took the opportunity because this was another powerhouse lab in Canada that was again doing edge research into this particular process of apoptosis. And they were generating all these gene-engineered mice, which was amazing because you could knock out a gene and find out what that did to an infection. So I went over there and it was an amazing time, 5 years, and then came back to head a lab and then eventually head a division at Weehai. And then just a year ago came up to Sydney to become the director of Centenary. So that's a little bit about the journey of my research. But what I wanted to do now is touch again on some anecdotes along the way. So in terms of where do you start, you've got to pick, if you want to do research, if you want to become a clinician scientist, if you're entertaining it, as I said, give it a go. You can't really make a big mistake here. Where do you start? Well, obviously you have to find a field of interest, and for me, it was always infectious diseases, but it just so happened that at that time, research was really flourishing in apoptosis or cell death. And I told you that even in undergrad, my favourite subjects were molecular biology or biochemistry. So this all made perfect sense to me when I started to sit down and think about it, and the opportunity arose to be able to go to Weehai and start studying apoptosis and apply it to my clinical interest, which was infectious diseases. The next thing you've got to do when you sort of know, well, this is the likely field I'd be interested in, and the likely field obviously could be much, much broader than that. So it could be clinical trials, it could be epidemiology, it could be global health, but my interest was very, very much fundamental science. You find a supervisor is the next step, and again I was pretty lucky because the Royal Melbourne Hospital is very similar to, is right next door to the Walter and Eliza Hall Institute. A very similar relationship between Centenary and RPA and University of Sydney. In fact, it was very identical. It was this little triangle, which we used to call the Parkville Precinct, which was Weehai and Royal Melbourne Hospital, and then University of Melbourne. So it was, we used to have sort of people from Weehai coming across to the hospital giving lectures. And one day Susan Corey came across and she gave a pretty nice talk on apoptosis, and I was completely hooked. So the first thing I did was to email her and say, I want to do molecular biology, clinician over here at Royal Melbourne Hospital. She wasn't too fond of clinicians, but she put me in touch with Andreas Strasser, who was fond of clinicians, strangely enough, primarily because he wanted to be treated for a whole lot of things, and his children as well. So he actually invited me around, we had a chat, and actually kicked it off. He was an amazing fellow and an amazing mentor along the way. So you just use your contacts and you find someone that might be good, ask people, particularly when you've got your field of interest. The other thing which is very, very tricky for clinicians, you've started on this journey of making a reasonable amount of money, and suddenly you have to contemplate this massive fall off a cliff. Because there ain't a lot of money in research, and particularly when you start doing your PhD. So I remember that when I first started my PhD with Andreas, every single weekend I was doing clinical research. So fortunately I didn't live too far away from Weehai, and I didn't live too far away from the hospital, so I was able to manage setting up my experiment on Saturday morning, rushing off seeing patients, and then Saturday evening coming back and the experiment finished. So it was all perfect. No, it wasn't. But anyway, I think that you've got to understand that yes, you do take a pretty big pay cut, but you do it because you like the work. Understanding the process really pertains to what paperwork do I need to fill out to do a PhD, and what do I need to get a PhD scholarship if you need a PhD scholarship. And you know that's usually dealt with by research officers at institutes or at universities. When you start your PhD, during clinical medicine, you'll have disappointments. People won't respond to therapy, but they're single individuals. When you've set up an experiment that's taken virtually a month to set up, and then you get the results and you go, your jaw drops because you sort of say, oh, there's nothing here. But then you pick yourself up and you say, but, you know, I actually enjoyed doing it up to the point when I got the result. So I'm going to do it again, but this time a little bit differently. And then eventually you'll get around to actually getting quite a bit of success. So I've supervised 10 clinicians that did PhDs or did masters. And if I went back to all of them, and many of them have been very, very successful. In fact, I'd say all of them have been successful, just in different ways. So of those that didn't pursue any further research, I sort of asked them, well, did you regret doing a PhD? And the answer is invariably no. They still enjoyed it because they had the experience and they found out they didn't want to do research. Well, that's still great. And then you still got this extra thing you put on your CV, which is called a PhD. So for the most part, it was very, very useful to them. For those that did continue down that stream, well, they've done incredibly well. Heads of department in gastroenterology, in ID and other areas. So it really was incredibly useful, which is why I sort of say try it, because you're unlikely to fail. Where to next? So for me, Andreas was a terrific supervisor, but he also later on became a quite important mentor for me in choosing the next place to go to a postdoc. And then when I did my postdoc with TACMAC, he became not just a mentor, but also a very, very good friend of mine who I keep in touch with to this day. In fact, I'm going over there for the 40th anniversary of the discovery of the T-cell receptor in a few weeks. So you really do need to pick up mentors along the way. And it doesn't have to be a formal relationship. I always try and get people to go into some sort of formal relationship, but it can just be an informal thing where you just pick up the phone, call people, or send them an email saying, I've come to a position where I have to make a decision, would mind some of your knowledge or wisdom to guide me. How much do you commit? So this is always a question, and it came up in the previous discussion. So when I first entertained being clinician-scientist, I said, I'll do 50-50. Sure, you can do 50-50. And then I looked for examples of this 50-50, and there was no real examples of 50-50. I didn't see anyone who was doing 50% clinical work, 50% research. You do what you like to do. So yeah, for me, it became 80% research, 20% clinical, and these days it's even a bit less than that. It's more like 90% research and 10% clinical, and that's just to keep my foot in that door. So it's really up to you what you decide to do, but I think that one of the nice things is the clinician scientist is that you can spend a little less time in the clinic but not lose your expertise. Either you've got that acumen and it comes to you naturally. In science you do have to do the hard yards, so you do have to read papers, you do have to keep up with the research, and no one's going to help you in science. In medicine, you'll always have people that give you a hand. And I always jokingly say that, you know, in the clinical sphere, when people say, Pellegrini, what are you doing? You don't know what you're doing. And I say, look, I'm an okay clinical physician, I'm an okay physician, but I'm a brilliant scientist, okay? And they say, oh, okay. And then in the scientific sphere, the reverse happens. They say, Pellegrini, what are you doing? That's a ridiculous design. And I sort of say, well, yeah, I'm an okay scientist, but I'm a brilliant physician. So just use that line, and you can really, really, really accommodate yourself in both spheres, because neither of them know what you're doing at the other place. It's so good. Then the other thing I wanted to mention is multitasking. Now, we all use that term, and I've got that little joker's hat there, because it is a little bit of a joke for me, because multitasking for me is doing 5 things adequately. Or maybe 4 things adequately and 1 thing pretty well. But it's never doing all those things or 100% of those things really, really well. So that's the first thing I sort of learned, is that you've got to realise that you can't do everything perfectly. So some things, when you're writing an email, sometimes I look back on an email that I've written and I think, my God, I don't even understand what I've written here. But presumably other people can sort of pick it up. So yeah, I don't proofread 10 times because I don't have time to proofread 10 times. But that's something that I cop. So yeah, I'm happy to not be a master at all of these things. I'm happy to participate in them all. Some of them I do well, some of them I don't do quite so well, but it's adequate. So for me, multitasking is very much around making sure that you're okay or adequate at everything you do, and particularly in the clinical setting, obviously. But the other thing is too, whilst you're multitasking, you prioritise things. So some things you sort of suddenly decide, well, this is something that I've got to do really well, so I'll put the other things on the side and do them in the meantime adequately. But this is the thing that I really, really need to concentrate on right now. And it could be writing a grant, or it could be writing a paper, or it could be doing an actual experiment or designing an experiment. So one of the first things will happen to most people when they undertake a PhD is this imposter syndrome. You just don't feel you fit in, particularly for clinicians or people that have got a medical background. You go into this lab, there's a whole lot of high-powered scientists, and you sort of think, what am I doing here? I don't know anything. And it's really, really important to understand that the PhD is a learning process. I call it an apprenticeship, so that's all it is. So you're going in there as an apprentice to understand how to do science. And whichever avenue you take in your PhD, it doesn't matter, because it's a real exercise in clear thinking. You design an experiment, you design a protocol, and you make sure you've got the right controls. You make sure that whatever you're going to deliver as a data is going to be interpretable and robust. So that takes time, and you never start as a leader, but eventually you can become a leader, and you'll get people helping you with experiments. You'll get people that suddenly want to come and do a PhD with you. The other thing in science, a lot of people get intimidated by competition. So you'll go to conferences, there are these big, phenomenal speakers, and don't be intimidated, because they've got probably their own sort of Achilles heels as well. And I think for the most part, a lot of scientists, very hardcore scientists, really appreciate having clinicians around, because ultimately, at the end of the day, they have to find some sort of something that makes their research relevant, and it has to be clinical, and it's really the clinicians that actually can guide, well, that research is not particularly... A number of times I've sort of had people come to me and sort of said, what do you think about this idea? And I said, how's that relevant to human health? And you sort of think, no, go away, think about it again. And so everything I do has always got that that magnifying glass or view as to focus is this clinically relevant? Can I envisage that this might be clinically relevant? So clinicians are always embraced by research, always, always. I think you do have to identify what your assets are as you're building up your career. What do you want to make your particular asset, distinguish you from everyone else? And you can usually do that. You build it and you acquire it. I think one of the first things, one of the biggest hurdles for me was moving from having done a postdoc in Toronto to being recruited back to being a lab head at Walter and Eliza Hall Institute. And I remember talking to one of my mentors in Toronto, and I said, this is incredibly frightening for me, because I've always been surrounded by people that help me, give me instructions, and I sort of think, I'm going to be all by myself. And they said, bullshit, you're going to be by yourself. You've got collaborators, you'll have PhD students who'll help you do the work, you'll have postdocs and recruit postdocs. And I sort of thought about it, and I thought, nah, I don't believe a word you're saying. I went and became a lab head, and it was all true. Everything sort of did fall into place, because suddenly you had a vision and you were able to create and bring other people along with you for that particular vision. So you do find your niche, and you do engage with people, and you've got to have that support. And to this day, probably the thing that I like most about science is talking to other scientists about my ideas and hearing their ideas, because science has now become so collaborative. And I said to you, even at the level of engaging more clinicians to understand how their research becomes relevant. So that interaction between people is incredibly, incredibly important, and that's really the salvation for my career trajectory, is that I just put myself in amongst really good people. Managing disappointments, it can be at the level of experiments, and I don't know why I put Richard Bagwell up there. He hasn't had a lot of disappointments, but I suppose he is a mentor. He's a mentor for Women's Franklin, so that's why he's there. But he's actually an amazing scientist. He does genetic heart disease, sudden death. You will get rejection, and you've got to become comfortable with rejection. And I don't know how many times I've been rejected by journals, by funding bodies, by everything. So you take it, you go through a grieving process. For me, that angry business lasts for a bit longer than most other people, but you're angry, you get over it, and you say a lot of rude things about, you know, NHMRC or whatever the funding body might be. But ultimately, you all calm down and you think, well, you know, that's the business, right? Think about challenging some rejections. So, you know, I get rejected from journals and papers, and you sort of write back to the editor and you say, well, you know, what's wrong? I've seen something else published which is much worse than my work. What are you talking about? And so you can challenge people about your rejection, but I think that the important thing is that you've got to listen to all the feedback you get, but don't necessarily act on it. Just take it all in. So if I responded to everything that I was told, I probably wouldn't get out of bed. So you just take it all in and digest it, and eventually you start to make some decisions about how you might want to act. But never act hastily. I've mentioned find a mentor. In fact, that should be on every one of these slides. Should be find a mentor. And I think just to distinguish, you'll have a supervisor. So if you do a PhD, you'll have a supervisor. A supervisor, I don't call a mentor. So Andreas became my mentor after I finished my PhD. All he wanted me to do was do all his bloody work and make sure I finished it, and then he wanted me to go to a lab so that he could get mice from another institution. So I knew all this, so that's why I had another mentor on the side who was able to give advice and sort of said, well, you know, as it turns out, his advice is probably okay. So do find a mentor. The other thing that's nice is that your mentor may or may not be a champion. So the other thing you've got to find is a sponsor or a champion. So it might be the same person, but it might not be. Because a champion is someone that really, really helps elevate you to the next level. Will actually try and help you along the journey, find opportunities for you. Why would someone want to do that? Well, it's funny, but I find myself doing it for many, many other people, because suddenly you sort of think, well, I really, really would like to live vicariously by seeing other people's career trajectory really, really become amazing. And so you're caught up in the moment, and you want to try and help other people who you think do have an amazing career in science or in the clinical realm or wherever. So yeah, that should be on all the slides, and I sort of think it is, but anyway. Managing expectations and visions. Like you're always going to, we talked about this in multitasking, but I sort of say to people, you just have a wish list without any constraints. Just think of the most amazing things you want to do, and it's not always going to work, and there will be constraints. You will have to make concessions. You might have to compromise a little bit, which is not giving up, it's just maybe compromising a little bit. But if you've got your list of big wish list and then you can prioritise it, then you can probably see a lot of it through. The other advice would be, what your wish list is today might not be your wish list tomorrow, and things do change. So be prepared don't be married to any single idea. Be prepared to surrender it and pivot if it's just not working for you, which is why I also say, do a PhD, try it, because you can pivot back and go back to clinical medicine. And as I said, some of the people that I've supervised have done exactly that, gone back straight into clinical medicine, and as I said, they don't regret doing their PhD. They actually quite enjoyed it. We love clinician-scientists at Centenary. In fact, we embrace it because obviously we're doing so much fundamental basic biology that we need to have that oversight of clinicians coming into us and saying, yes, that's relevant, or yeah, no, that's not relevant, or let's see how this could be relevant. Because some of our work is just so basic, you don't really know if it's going to be relevant, and you need a clinician to sort of come along on the journey for you. So we're going to be part of, when I say we, it's the Centenary Institute will be part of the Sydney Biomedical Accelerator, which is a massive building, multi-buildings going up in the, behind, or actually between University of Sydney and RPA, which is, sorry? The meet again. So this is an amazing set of buildings which will house, I think, about 1,500 scientists and researchers. The whole remit is to accelerate research, which is to bring academics from the university, translational scientists, basic scientists from Centenary. We are the only MRI at the moment that's involved in the partnership, and the clinicians from the hospital. And I've said, if you want a position in there, you've got to bring those two partners along with you to give relevance for any program of research that you take there. My last slide. Will you try? Thanks.
Thanks very much, Mark. Now, do we have some questions? Louise, you've always got one.
Louise Bauer, President of the Academy. Thank you so much, Mark. Yet another fantastic talk in a different series, but also related reflections. Mark, could you just tell us a little bit more about how you got funding to do your PhD, and when in your training? So you did medicine, you then presumably did physician training, and then somewhere along that line, you must have taken some time out to do a PhD. When did you do that? How was your PhD funded? Could you tell us a bit about that?
Yeah, so I got my FRSCP, well, and probably the day I got my FRSCP, I went into the lab and started doing my research. So as soon as I got my FRSCP and became a consultant, I started to look for opportunities, and I learned very quickly that you have to apply for a scholarship. So I went through NHMRC and applied for a PhD scholarship, which is available primarily to clinicians to do research or PhDs. So I was very lucky to get one of those scholarships, and so I went straight into the lab. So there was absolutely zero time between me becoming a consultant. I think I was a consultant for maybe 2 months before I went straight into the lab. But the wonderful thing is, because I was a consultant, I could continue doing quite a lot of clinical work whilst I was doing my PhD, which helped fund my research. So it was just luck that I finished training, went straight, found the job that I wanted. I probably looked about a year earlier. That's the only trick I'd say, is that if you're contemplating this, you probably need to give yourself a year to understand exactly those aspects of how do I get funding, do I need funding, will someone support me? So University of Sydney will certainly offer a lot of scholarships. So there's lots of avenues to fund a PhD scholarship, and lots of avenues to get top-ups as well.
Mark, most Australian clinician specialists and also researchers do have periods overseas. How do these young people think about where they might want to go overseas?
Yeah, and I think that you could either do your PhD overseas or you can do your PhD here. In terms of the landscape's changed a lot. So I always used to tell people that did a PhD, you've got to go overseas. You've got to broaden your horizons. You've got to go overseas. You've got to form these collaborations. I don't think that's true anymore. I think you can stay here. You probably need to change and swap it around and maybe go to a different city or somewhere else. But I think the opportunities here are great. I think the research here is really, really good. The only reason why I'd suggest, and this is probably not a great reason to be telling people, but go overseas because it's a period where you can travel. Like you can really travel and explore things. It's, if you're doing it as a part of your PhD, if you're doing it as a postdoc or an MPH or whatever you want to do, it's a real opportunity to travel. And so Toronto was great for me because we didn't have kids at that stage, so it was great. So we would go on these trips to New York, which was only a half-hour plane. We would go to Grand Canyon and do all these things, and then our first kid was born, and that's when we decided we better come back to Melbourne.
Yeah, it's a bit like Anushka says, enjoy the journey along the way and make the most of it. Are there any other questions? Okay, I've just got one last question. Where did you get that shirt and those shoes?
Can't tell you. No, they're all Italian.
Great. I'm glad Italians recognise that Australia's centre of the world then. Okay, well, thanks very much, Mark. That was great.